Welcome to True Projects. In this video, we are going to explain about the project that is our AI net, tomato plant disease classification using residual attention inception network. So before getting into the execution, first let us understand what is this project about. Tomatoes. Tomatoes are one of the most important crops that we grow, not just because they are widely eaten, but also because they support farmers and also contribute a lot to the agricultural economy. The problem is that tomato plants are very prone to diseases that are caused by bacteria, fungi, and viruses. These usually show up on the leaves as things like curling, yellow patches, spots, or blight. When diseases spread, they can wipe out a large portion of the harvest, sometimes up to 40%. And this means big losses for the farmers and also for the economy. So, this problem becomes even more serious when we think about the future. By 2050, the world's population is expected to reach nearly 10 billion, which will push the demand for food even higher. At the same time, challenges like global warming, changing weather patterns, and the faster spread of pests and pathogens are making farming harder. That's why protecting tomato crops is so important, not just to save yields today, but also to ensure we can meet growing needs food that is growing food needs for the future. So traditional tomato plant detection models, that is traditionally tomato plant diseases, they are identified by farmers and agricultural experts through direct observation of the leaves. While this approach can work, it takes a lot of time, requires significant effort, and can be inconsistent because it depends heavily on the human judgment. This makes it impractical for large scale farming or for detecting diseases at an early stage when quick action is most important. So, machine learning models have also been explored, but they often rely on manually extracted features and struggle to maintain accuracy under different conditions, such as changing lighting or leaf appearance. Because of these drawbacks, there is clear need for solutions that are accurate, efficient, and scalable. Such systems would make it possible to detect diseases early, reduce yield loss, and also ultimately support food security. So to overcome with all such issues, the proposed system it uses deep learning model called residual attention inception network, that is RAI net, to automatically detect and classify diseases in tomato leaves. At its core, it builds on a pre-trained ResNet 18 model, but instead of training the whole network from scratch, only the last two layers are retrained. This makes the system faster to train and more efficient, while still benefiting from the knowledge ResNet 18 that has already learned from large image datasets. And to make the model focus on the important parts of the leaf that show disease symptoms, a channel attention mechanism, that is, squeeze and excitation block, is added. This module works by giving more importance to useful features while reducing the effect of irrelevant ones. In simple terms, it helps the model to pay closer attention to disease related patterns, even if they are very subtle, which improves its ability to correctly identify different types of disease leaves, that is, leaf diseases. And also the model includes Inception module, which uses different sizes of filters to look at the leaf images in multiple ways. This means it can capture both the small details like tiny spots or blights as well as large patterns like curling or yellowing across a whole leaf. After analyzing these features, the model makes its final decision using global average pooling, followed by fully connected layers, which combine all the learned information to predict the disease classes. And to help users to understand how the model make its decision, GradCam is used. This technique highlights the specific areas of the leaf that the model focused when identifying a disease, make the predictions more interpretable. Moving on, we have also added a detection module as the extension. So classification will be just telling which type of disease the tomato plant has. So in detection, it will be localizing the area where that disease is present. So for that, we have used YOLO model, which allows it to not only classify the disease, but also pinpoint the exact areas of the disease that are affected. This means farmers or users can see exactly where the disease is on the leaf rather than just knowing what type of disease it is. Furthermore, to make the system easy to use, Flash-based framework has been created. That is, a user-friendly web interface has been created using Flash framework. Through this interface, users can simply upload an image of tomato leaf and get predictions from trained models. This makes the system interactive and practical use, allowing farmers to quickly detect and understand leaf diseases without needing technical expertise. Moving on to the implementation of the project. To implement this project, we have followed the steps which are mentioned in the flow of work. So let's understand what is the procedure followed. So first we will be importing all the required libraries. So these include tools for image processing, deep learning, model training, and visualization. Then comes data preparation. So here the new plant disease dataset of tomato leaf is collected for the classification. The images are labeled according to the type of disease. And for the YOLO based detection, bounding box annotations are prepared in YOLO format, that is TXT format, to mark the abnormal regions. The dataset is then divided into training validation and testing sets for model development and evaluation. Then comes visualization. So here, to better understand the data set, the distribution of the classes is plotted using bar graphs. Sample images with their labels or bounding boxes are displayed to verify that annotations are correct and consistent. Then comes pre-processing. So here at this stage, all images are resized, normalized, and augmented to improve model training. Torch vision transformers are used to pre-process for the classification. For YOLO, a data.yml file is created containing datasets paths and class names. Two parallel datasets are maintained. One for classification, that is with the images as well as labels, and another for YOLO detection, that is image with bounding box labels. And moving on to training, all the classification models are used to train the dataset for classifying diseases in the tomato leaf, and for detection, we have used YOLO based models. And they are evaluated using their respective metrics. So, for classification, we have used accuracy, precision, recall, and FN score. And for detection models, we have used mean, average, precision, precision, and recall. And we will be saving best version, that is, best performing model for future prediction. Coming to inference pipeline, here, new image will be given to the system. The pipeline includes two outputs, a bounding box highlighting the abnormal regions and the predicted disease class of the leaf. And coming to the part of front end. So the user interface is developed using Flask framework. It includes user registration and login functionality with an SQLite database to manage users and their sessions. And users in the front end, they will be giving the input. If they want classification, they will get classification. If they want detection, they will be getting detection. That will be irrespective of their needs. So that will be pre-processed at the backend. It will be fed to the train model, and that train model will be giving us a prediction. So this is how we have implemented the project. Moving on to the algorithms used. So these are the algorithms used for classification, and these are the algorithms used for detection. And coming to the requirements, to implement this project, basically we need hardware and software requirements. In hardware, we need operating system of Windows, processor of 5 and above, RAM of 8 GB and above, and hard disk of 25 GB and above. And in software, we need Python application, front-end framework of Flask, backend framework of Jupyter Notebook, database of SQLite 3, and front-end technologies are HTML, CSS, JavaScript, and Bootstrap 4. So this is the overview of the project. Now let's execute. To execute, open the code folder that contains source code files. And these are the source code files in the code folder of the projects. Let's understand what are these. And here in the model folder, we have the algorithm information that will be loaded into the project code during runtime of the application. And in this, we have dataset. So let's open. Here we have dataset in two subfolders, that is train and valid. Let's see in train. Here we have different types of tomato leaf diseases in different folders. In this folder, we have bacterial spot tomato leaf disease. And in this, we have early blight. And in this, we have healthy tomato leaves. Accordingly, we have diseases of tomato late blight, leaf mold, specterial, that is septoria leaf spot, spider mites, target spot, tomato mosaic virus, and yellow leaf curl virus. So these images are used for classification. And images which are in the train folder will be used for training the model. Similarly, we will be having images in the validation folder also, but they will be used to evaluate the model. And now get back. So this is the static folder. This folder consists of files related to CSS, JavaScript, and Bootstrap files, which are important for the visual appeal. And this is the templates folder. This folder contains all the HTML pages used in this project. It typically includes files like index.html, about.html, etc., which represent different pages of the website. And this is the test cases folder. This folder consists of the images that we will be using in the front end for doing prediction and detection. And here in this folder, we have images that are used for detection. So let's open train folder. Here we have images and annotation files. So in this, we will be having images, and in this, we will be having the object locations, that is the disease locations of the images. Let's open image folder. So here we have all different types of tomato leaf disease images clubbed together along with the healthy images, healthy leaves that is. Get back. And the disease locations will be in this label folder. Let's open this label folder. So this contains image annotations, that is disease annotations, or we call it as labels for the objects in corresponding images. Here, each label file, it corresponds to an image file in the image folder. For example, the first label file, it corresponds to the first image which contains the disease locations. Accordingly, for the second image, we have second label file. Accordingly, for all the images, we will be having all the annotation files. And these label files, they follow the YOLO format. Let's open file for sample. So here, this line represents a detected object. This is one type of class, that is one type of disease, and these are the bounding box coordinates. This is the X limit, Y limit, width, and the height. Close this. Same we will be having in all the files. Now get back, get back. So this folder is used to train the detection models to detect the tomato leaf disease. And here also we will be having images that are used to evaluate the model. Now get back. This is the app file. So this file contains the information related to front-end logic. It includes code written in Python that handles server-side operations such as processing user requests, interacting with the database, and generating dynamic content to be rendered in HTML templates. And this is the best.pt file. So this file contains the algorithm information related to detection model. And these two files are the Jupyter Notebook files, which contain a combination of code, graphs, and outputs all in one place. It allows users to write and execute code in individual cells, making it a popular choice for the data science. So in this Jupyter source file, we will be writing the code for doing classification. So we will be loading this dataset and train all the models for doing classification. And in this Jupyter Notebook file, we will be loading dataset which is in this folder and train all the models for doing detection. And this is signup.db file. This file is a database file which is used to store the user information. So this is all about the code folder. Now let's execute. To execute, copy the path of the code folder from the address bar of the file explorer. So I'm copying it. Now open Anaconda prompt. It is open. Give the command cd, give the space, paste the copied path. Now click on enter. Here, if you observe, current directory is changed to the code folder's path. So now compile app.py file. So give the command python space app.py and now click on enter. This command will execute Python script and perform a runtime check for any syntax errors or logical issues. And after running the app.py file, Flask framework will host the application locally at the default address, which is the local host and the port, unless configured differently. So this is the address where application has been hosted. Now copy this local link provided by the Flask framework and paste it into any web browser. I've copied the link. So paste it into any web browser. I prefer Chrome. I will paste it in that. Paste the link here and click on enter. So this is the web page of the project, which is displayed in the browser, and this is developed using Flask framework. So now we need to get signed up first. So click on sign up and enter all the details to create an account. I have already created an account, so I will directly sign in. Click on sign in, give the username and the password. Click on sign in. We have signed in successfully and redirected to prediction page of this project. So first we will be doing classification. Here we can choose file or either we can drag and drop the image. So let's click on choose file to do the classification task. Click on choose file and now open test cases. Open classification because we're doing classification now. So open any folder and select any image of your choice. So let's try with the first folder and select this image. Click on open and now click on classify image. So this given image, it will be pre-processed at the backend. It will be fed to the train model and then train model will be giving us the prediction. So this is the uploaded image, and here we have the analysis results that it is predicting tomato bacterial spot. So it is the correct prediction. It is predicting it correctly. So this is predicted by RNet model, and this is predicted by DenseNet model. So here we have scores also. So this is the score of RNet model, that is 99.53, and here we have DenseNet model score, that is 100%. And here this is the original image that we have uploaded, and this is the heat map visualization that is created with the GradCam. So here you can observe red and yellow color lines, right? That is red color and yellow color area. So this area, based on considering this area only, it is predicting this disease, that is tomato bacterial spot, that is tomato plant is suffering with a bacterial spot. And this is AI attention overlay graph. This is the image. I mean, this is the leaf, and this is the area where it is focusing on to predict this bacterial spot. Now let's try with some more images. And now click on choose file. Let's get back. Open this folder, select this and classify it. And this is the uploaded one. This is the AI attention heat map, and this is the AI attention overlay. This is general GradCam, and this is on specifically leaf. And this is the area that where it is focusing on. And here we have correct prediction that it is early blight. This is the score of RNet, and this is the score of DenseNet, and both are predicting correctly. Get back, click on choose file, get back. Let's try with this image. Now click on classify. So here it is proven to be healthy, that it is predicting it as healthy. Click on try another. Click on choose file. Get back. Open this folder. Select this one and classify it. So here if you observe, this is the original one, and this is the AI attention heat map. This is the AI attention overlay. And here we have exact prediction that it is late blight. Tomato plant is suffering with the late blight disease. Click on try another. Click on choose file. Get back. Let's try with this one. So here we got the prediction that it is leaf mode. Click on choose file. Let's try with this image. And here we got the prediction that it is septoria leaf spot. The tomato plant is suffering with the septoria leaf spot. And this is the score of RANet, and this is score of DenseNet. Click on try another. Click on choose file. Get back. So let's try with this one. And here we got the prediction that it is tomato spider mites 2 spotted spider mititis. Let's try with one more image. Click on choose file. Get back. Open this one. Let's select this one. And here it is predicting it as target spot. Now let's try with one more. Get back. Open it. And now let's try with this one. So here we got the prediction that it is tomato mosaic virus it is. And now let's try with the last image for classification task. Click on choose file. Get back. So let's try with this one. And here we got the prediction correctly that it is tomato yellow leaf curl virus it is. So we're done with the classification. As of now, you have just seen what the disease that tomato plant has. Now let's spot the area where disease is present in the leaf. For that, we are supposed to do the detection. So open detection. Click on choose file. Get back. Get back to test cases. Open detection. So you can select any image of your choice. Let's open this one. Click on detect image. So here if you observe, tomato early blight has been detected and a blue color bounding box is drawn around it and labeling it as tomato early blight with the 0.85 detection score. That is a probability score that is predicting how confident it is in that class. Click on try another. Click on choose file. Now let's try with this one. And here you can observe tomato leaf mold has been detected with the 0.98 probability score and a green color bounding box is drawn around it. Let's try with two more images. Click on choose file. Let's try with this one. So here this leaf is healthy and it is drawing a bounding box around it. Click on choose file. Open detection folder. And now let's select this one. Click on detect image. So here also early blight has been detected. Click on try another image. Now let's try with this one. Here tomato is healthy. Click on try another. Click on choose file. Let's try uploading from the classification folder. Open it and let's try with this one. So let's upload this one. So here you can observe application is correctly detecting tomato yellow curl leaf. Let's try with the last image. Click on choose file. Get back. Let's try with this one. So exactly it is detecting septoria leaf spot also. In this way, the application does the detection. Now let's check with the graphs. So under graph, click on classification. So here we have model comparison graph first. Here on x-axis, we have all the models that are trained and on y-axis, we have accuracy score. Click on close. And this is accuracy. And here we have model performance. And this is precision score. This is recall score. This is FN score. Close it. Now let's check with the detection graphs. Click on detection. So here we have precision score. On x-axis, we have all the YOLO models and on y-axis, we have precision. Close it. Here we have recall score. Here we have mean average precision score. Now close it. Click on sign out. In this way, just by uploading images to the application will be accurately classifying as well as detecting which type of disease that tomato plant has by using deep learning models and the YOLO models. This is all about the project.
Thank you for watching video. For more projects please visit our website www.trueprojects.in. For updates on latest project videos, please visit True Projects YouTube channel and subscribe.